I'm Adrianto. I'm the Director of Bioinformatics Engineering at Genomics England. And today I will share with you how our platform called Genie utilizes Nextflow and Seqera Platform to empower our generation study initiative. Let us revisit briefly the Genomics England journey. The 100,000 Genomes Project was launched in 2015. This was the world's first largest national sequencing program, and Genomics England was formed to guide it in partnership with the NHS. In 2018, we finished sequencing our 100,000 participant, and 2 years later, in 2020, we established what's called the Genomic Medicine Service in partnership with the NHS, essentially making NHS a national healthcare system that offers whole genome sequencing as part of a routine care for cancer and rare disease. And in 2021, we announced the Generation Study Initiative alongside several others. So this is our infinity loop. Genomics England has two linked functions. The first one is to support an evolution of genomic healthcare, and the second is to accelerate genomics research. So in order to do this, we work with the NHS to provide genomic testing so that the doctors can diagnose and treat cancer and rare disease conditions in the patient. And with the patient's consent, we provide this data in our research environment for our research users to essentially play around with the data and gain insights or kind of treatment that we can pass back to our NHS patients. As of May 2023, Genomics England has sequenced over 150,000 genomes. This corresponds to about 65 petabytes of data. 50% of the participants with cancer received a crucial diagnostic result and had their treatment plans altered based on the insights found. And there's more than 6,000 participants with rare conditions that received a diagnosis that had not been possible through routine care. In terms of research, we have about 135,000 whole genome sequences in our national genomics research library. We also have about 60,000 digital pathology images to help us boost our multi-omics and multi-modal capabilities. And we partner with 8 out of the 10 top pharma. So let me talk briefly about the Genomic Medicine Service. So this is a national scale ISO-accredited live clinical service that we provide in partnership with the NHS for cancer and rare disease. As I mentioned, this offers whole genome sequencing as part of the routine care. And we look at more than 30 indication for rare disease. For cancer, we look at acute leukemia, pediatric tumors, sarcomas, and several other complex cancer types. So this brings us to Generation Study. So every year, hundreds of babies in the UK are born with rare genetic condition. Early intervention can dramatically improve their health outcomes and quality of life. But timely diagnosis remains challenging and often delay critical care. So there's 3 objectives that we want to do in the Generation Study. The first is to evaluate the feasibility and benefits of using whole genome sequencing to screen newborns for a large number of rare childhood-onset genetic disease. The second is to understand how this genomic data can drive groundbreaking discovery, fostering development of new treatments and diagnostics for NHS patients. And the third, we also like to explore the broader implication of storing an individual's genomic information from birth throughout their lifetime, evaluating ethical dimensions alongside with the scientific one. So the generation study aims to sequence and analyze 100,000 genomes of newborn in the next couple of years. We seem to like this number in Genomics England. But we expect to see about 1,000 condition-suspected results during the study. So this equates to about 1%, essentially. And currently, at the moment, I think we estimate about 9 children born each day in the UK with a rare, treatable rare condition. So what does this mean in terms of diagnostic discovery? Sorry, diagnostic odyssey. This is something that we are essentially trying to target as part of the generation study. So traditionally, when a baby is born and they develop symptoms, then they get referred to a specialist. They had to go multiple tests before they can get a diagnosis. This could take some time, essentially. And like I mentioned previously, time is crucial in this condition. What we have in the Genomic Medicine Service, in a way, we try to simplify the test. So rather than having multiple tests for the newborn, you go for, or the patient, you have a single genomic test, and then they can have the diagnosis. However, as you can still see, this means that the baby or the patient still needs to develop symptoms first before they can get referred to a specialist. This is kind of reactive, so we want to move on to something more proactive. And essentially, in the generation study, we want to skip the middle bit. When the baby is born, we do a kind of like a preemptive screening, and if there's a diagnosis, then the baby and the parents can be informed. So the key here is essentially early intervention, timely diagnosis, and timely treatment. So where we are at the moment, as of April 2025, we've enrolled over 9,000 parents. We've processed about slightly more than 5,000 cases through our pipelines, and we've returned 25 condition-suspected results back to the NHS. Additionally, we have slightly over 200 cases with prioritized variants under clinical science review. This is across 28 sites across the UK. So, how is this being delivered? Historically, our bioinformatics workflows are being run by something called Bertha, which is our in-house built workflow manager. It was built in 2014 as part of the 100,000 Genomes Project when tools like Nextflow haven't existed or not yet widely adopted. It's a registered medical device. We use it to deliver the 100,000 genomes, and it's currently also powering our GMS pipelines. The name, if you want to know, of Bertha comes from a 80s TV character. It's a machine, essentially, that can produce anything you want, but they tend to encounter difficulties along the way, which is a bit of an apt name. So yeah, we do have challenges with Bertha, because I think although it's effective in delivering the 100,000 Genomes Project, there's certain aspect, for example, the business logic and the orchestration were tightly coupled. It's not really modular. There's a lot of dependencies. Therefore, when we have a new case, a new use case, new initiative, it's quite difficult to adapt. It doesn't use kind of industry standard tools. So when a new joiner joins the team, the learning curve is quite steep. There's concerns in terms of scalability as well, because it's not really compatible with kind of cloud or hybrid storage and compute. And last but not least, it doesn't have a user community outside of Genomics England, which means we can't take advantage of what the wonderful community has to offer, like NF-Core or NF-Test. So in contrast, Genie represents kind of like our next generation solution. It's designed to be modular. We decouple the pipeline code from the orchestration and the business logic. It's adaptable to new use cases. It also uses kind of industry standard tools for ease of adoption. Last but not least, it's scalable and compatible with cloud compute and storage environment. And Genie under the hood, essentially, as we can see in the next few slides, is powered by Nextflow and Seqera Platform, which has these principles and features out of the box. So what does Genie do? At a high level, Genie efficiently manages workflow events. So from receiving genomic data to, let's say, do variant calling or maybe prioritizing those variants for clinical review. So it leverages a Seqera platform to execute Nextflow workflows seamlessly in various different computational environment. In practice, what this does is when an event occurs, so this can be, for example, genomic data being delivered or previous workflows finished running, this will kind of trigger an event. The event will kind of trigger Genie. And yeah, Genie decides which workflow it's going to launch based on the trigger. A launch workflow in Seqera will essentially be received, and it will select kind of the correct workflow from the repository. Seqera Platform will then run it in the environment that we have selected for it. Once it completes, it will essentially raise an event. It goes back, kind of completes the cycle, and it'll continue until there's no more workflows to be run end to end. But the idea is essentially to build a generic system that's able to deal with launching any sets of Nextflow workflows on any compute environment in response to any event. So looking forward, our plans is to scale up the generation study. We expect to see a significant ramp up in the sample processing to help us get to 100,000 in the next couple of years. We're also in the process of refactoring our Genomic Medicine Service workflows to Nextflow native implementation. And I think once that's done, we'll migrate fully from Bertha to Genie. We'll also look at kind of the cloud compute and storage to provide resiliency and also the scalability. And this will help us to set up the exciting use cases in the future of what we'll have in Genomics England. Thank you for listening.
Thank you, Adrianto. Okay, so I've got a question here from Sean. Alongside whole genome sequencing in adults, are other health data and medical records collected too that will help studies of single nucleotide polymorphisms or aid GWAS in some way?
Yes, so our research environment also includes clinical data. The data is obviously de-identified, but there are other types of data available in the research environment. I think I've also mentioned imaging data. We have some transcriptomic data as well. So there are different types of kind of data available for our research users to look at it, essentially.
So it's really, I guess you just think about, especially in newborns, you said 1% chance of possibly, was it a rare disease? What proportion of those are novel and what proportion are unknown?
I don't have the answer at the moment. I'm happy to go back to the team and answer that potentially.
Yeah.
But it is essentially still ramping up. Like you can probably see in the sites, we currently have 25 conditions suspected that's being reported to the NHS. So there's still a long way to go before we reach the 100,000. As you're aware, in the newborns, it takes time because once you're recruited, it normally takes up to 9 months before you can actually get the sample. There's nothing we can do to speed up the process. So it's still a work in progress, I guess, in that sense.
Cool. And there's some good success stories for this as well. I think with ONT and maybe working in collaboration with Genomics England, possibly had some studies where the turnaround times on going from getting sequencing data all the way to actually diagnosing an issue, possibly leading to treatment, can have a huge effect. Cutting down those times. So it's super useful. What on average, in terms of the actual pipeline that you're running on the infrastructure, how long does it take to run from end to end? As soon as you have an event that you've received some data to get all the way to the end where you can then feed that information back to the clinician?
I need to double check just to make sure. I have a number in mind, I think. I know for the GMS, rare disease, for example, I think on average it takes about 10 hours for the pipeline. But for the newborn, I need to double check just to make sure. Because I think for the GMS, it tends to be more complicated because there's cases where you look at trios, so the parents as well. For the newborns one, I need to double check.
But it's still on the order of days rather than months or
it's definitely
that we had
months.
before, right? So it's great work. Yeah. Okay, I think we're just about on time.